Chapter 3: Innate Immunity. The induced response to infection. This flowchart summarizes the stepwise progression of the immune system's response to infection, beginning with the innate immune system and transitioning to the adaptive immune response if the pathogen persists. The first line of defense is the immediate innate immune response, which occurs within 0 to 4 hours of pathogen invasion. During this phase, the pathogen proliferates, but it is recognized by performed soluble effector molecules and resident immune cells in the infected tissue. If this response is successful, the pathogen is eliminated quickly, the infection ends, and only very minor tissue damage needs to be repaired. If the pathogen is not eliminated at this early stage, the body initiates the induced innate immune response, which spans from about 4 hours to 4 days. The pathogen continues to proliferate, and resident immune cells in the infected tissue become activated. These cells then recruit additional effector cells, leading to inflammation, fever, and the acute phase response. The recruited soluble effector molecules and immune cells attack the pathogen. If successful, the pathogen is cleared, the infection ends, and minor tissue damage is repaired soon after. Should the pathogen still persist beyond this point, the immune system escalates to the adaptive immune response, which typically begins after 4 days and continues until the pathogen is either defeated, the host succumbs, or the infection becomes chronic. The adaptive phase begins when secondary lymphoid tissues near the site of infection detect the pathogen. In response, pathogen-specific B and T cells are activated, proliferate, and mature into effector cells. These effector cells then migrate to the site of infection. If the adaptive response succeeds, the pathogen is eliminated, the infection resolves, and major tissue damage is gradually repaired. In the unfortunate event that the pathogen is not eliminated by the adaptive response, the host may die from the acute infection or may endure the long-term consequences of chronic disease. Cellular receptors of innate immunity distinguish non-self from self. This image illustrates how the innate immune system differentiates between self and non-self using specialized cellular receptors on immune cells. On the left side, a macrophage is shown recognizing a bacterium. The macrophage's surface is covered with receptors that identify specific carbohydrate patterns unique to bacterial cells. These structures are not found on human cells, allowing the macrophage to target only invading microbes while sparing the body's own tissues. The image highlights this distinction by labeling self-structures with a different shape and orientation than those seen on the bacterium. On the right side, the focus shifts to a natural killer cell. These cells monitor the surfaces of human cells and can detect changes that occur when a cell becomes infected with a virus. Virus-infected cells often present abnormal or altered surface proteins, which the natural killer cell recognizes using its receptors. Once detected, the natural killer cell releases granules containing cytotoxic molecules that lead to the destruction of the infected cell, helping to control viral spread while leaving neighboring uninfected cells unharmed. Together, these mechanisms demonstrate how innate immune cells can accurately identify and respond to infected or foreign threats without damaging the body's own healthy cells. Tissue macrophages carry a battery of phagocytic and signaling receptors. This table presents the various types of receptors found on the surface of tissue macrophages, which are immune cells that act as sentinels throughout the body. Each of these receptors plays a distinct role in identifying and responding to invading microorganisms. The mannose receptor, known more formally as cluster of differentiation 206, assists macrophages in engulfing bacteria by recognizing specific sugar molecules found on bacterial surfaces, including lipopolysaccharide, capsular polysaccharides, and manocylated lipoarabinomannan. Complement receptors identified as molecule 1 in cluster of differentiation 11b and 18 are also involved in engulfing microbes. These receptors detect sugar chains and proteins on both bacterial and fungal cells, particularly beta-glucans, which are complex sugars present in fungal cell walls. The receptor known as dectin-1 is especially important in recognizing fungi, although it also binds to certain bacteria. It detects sugar molecules such as beta-glucans and ligands from the cell walls of mycobacteria. Another receptor, called the macrophage receptor with collagenous structure, assists in bacterial recognition and engulfment by binding lipopolysaccharide and proteins found on the pathogen surface. The scavenger receptors, which are divided into type A and type B, bind to a variety of bacterial components. The type A version detects lipopolysaccharide, lipoic acid, proteins, and certain bacterial genetic material rich in cytosine and guanine. The type B version recognizes a type of lipid protein molecule known as diacylated lipopeptide. Cluster of differentiation 14 is another surface receptor involved in engulfment. It binds a broad spectrum of bacterial components including peptidoglycan, lipopolysaccharide, lipoic acid, and mannuronic acid. Finally, toll-like receptors form a family of 10 distinct receptors that do not participate in engulfment but are essential for signaling. These receptors detect many different molecular patterns from bacteria, viruses, and fungi and trigger an appropriate immune response. Together, this collection of receptors enables macrophages to both recognize and respond to a wide variety of pathogens, either by engulfing them or by initiating defensive signaling pathways. This illustration shows how tissue macrophages use various surface receptors to recognize, bind, and eliminate harmful microorganisms. In the first panel, several specialized receptors line the macrophage membrane. These receptors detect unique structures found on the surfaces of microbes. The mannose receptor and dectin-1 are able to detect sugars found on bacterial and fungal cells. Complement receptors detect molecules that have been tagged for destruction. Scavenger receptors, including type A and type B, as well as the macrophage receptor with collagenous structure, identify microbial patterns like lipopolysaccharides and proteins. Once a bacterium or yeast cell makes contact with these receptors, the macrophage binds the invader. The process continues as the macrophage wraps its membrane around the microorganism, internalizing it into a membrane-bound compartment called a phagosome. In the next phase, the phagosome travels deeper into the cell where it fuses with a lysosome, a structure filled with destructive enzymes. The result is the formation of a phagolysosome. Finally, inside the phagolysosome, the microorganism is broken down and neutralized by the enzymes and reactive substances released from the lysosome. Through this coordinated series of steps, the macrophage eliminates invading microbes and prevents them from spreading infection. Recognition of LPS by TLR4 induces changes in macrophage gene expression. This diagram shows the basic structure of a toll-like receptor, which plays an essential role in the innate immune system. Toll-like receptors are embedded in the cell membrane of immune cells, such as macrophages. They act as sentinels that detect signs of infection. At the top, facing the outside of the cell, is the region responsible for recognizing molecules commonly found on the surface of pathogens. This is known as the pathogen recognition domain. It identifies unique microbial features and alerts the immune cell to danger. The curved yellow structures shown in the diagram are these extracellular domains. When they detect a microbial molecule, such as a component from a bacterial wall, the receptor becomes activated. Beneath the membrane, inside the cell, is the region responsible for relaying the signal. This internal section is called the Toll/interleukin-1 receptor domain. Once the receptor is activated by the presence of a pathogen, this internal region triggers a cascade of cellular signals. These signals ultimately change which genes the macrophage expresses, prompting the production of molecules that help combat the infection. In short, this receptor detects harmful microbes outside the cell and rapidly informs the interior machinery of the immune cell to mount a defensive response. This diagram explains how a macrophage, an important immune cell, detects the presence of bacteria using a specific group of proteins embedded in its surface. When a bacterium is nearby, it sheds a small molecule called lipopolysaccharide, which is a component of its outer membrane. This molecule is recognized as a danger signal by the immune system. First, a helper molecule called lipopolysaccharide binding protein grabs the lipopolysaccharide and brings it to the surface of the macrophage. There, the lipopolysaccharide is passed to another protein called cluster of differentiation 14, which helps hold it in place and present it properly. Next, a toll-like receptor on the surface of the macrophage binds to the lipopolysaccharide. This receptor does not work alone. It needs help from a partner protein called myeloid differentiation factor 2, which fits into the toll-like receptor and stabilizes the entire complex. Once the lipopolysaccharide is securely bound to this group of proteins, toll-like receptor, myeloid differentiation factor 2, and cluster of differentiation 14, the complex becomes activated. This activation triggers the macrophage to respond by altering its gene expression and beginning to produce signaling molecules that alert the immune system and promote inflammation. In this way, the macrophage uses a team of proteins to sense the presence of bacterial invaders and kickstart an immune defense. This image illustrates how a bacterial infection triggers an immune response inside a macrophage, a type of white blood cell. When a bacterium releases a specific molecule called lipopolysaccharide, a protein on the surface of the macrophage recognizes it. This recognition happens through a complex formed by toll-like receptor 4, myeloid differentiation factor 2, and cluster of differentiation 14. Lipopolysaccharide binding protein helps deliver the bacterial molecule to this complex. Once the bacterial molecule is detected, a signal is sent inside the macrophage. This signal starts with a protein called myeloid differentiation primary response JON88 binding to the toll-like receptor. This interaction activates other proteins inside the cell, beginning with interleukin-1 receptor-associated kinase 4. This kinase activates another protein called tumor necrosis factor receptor-associated factor 6, which in turn activates a set of proteins called a kinase cascade. One of the important proteins in this cascade is inhibitor of nuclear factor kappa B kinase. This kinase phosphorylates another protein called inhibitor of nuclear factor kappa B, marking it for degradation. When this inhibitory protein is removed, nuclear factor kappa B is released and moves into the nucleus of the cell. Once in the nucleus, nuclear factor kappa B activates the transcription of genes that encode inflammatory signaling molecules. These molecules, known as cytokines, are then produced in the cytoplasm and released from the cell through the endoplasmic reticulum. The released cytokines help alert and recruit other immune cells to the site of infection, beginning the inflammatory response. This image shows a young child with visible signs of inflammation in the gums, particularly around the front teeth. The image appears to relate to the physiological consequences of immune dysregulation, possibly a clinical case tied to a mutation in the toll-like receptor signaling pathway. In the context of the previous diagrams discussing toll-like receptor 4 and its role in recognizing bacterial lipopolysaccharide, this child may be exhibiting symptoms of a genetic defect in this pathway. Such defects can lead to impaired recognition of bacterial components and reduced cytokine production, which compromises the body's ability to mount an effective innate immune response. In clinical immunology, patients with such defects often present with severe, recurrent infections or abnormal inflammatory responses. This image serves as a real-life illustration of what can happen when key immune signaling pathways, like those initiated by toll-like receptor 4, are disrupted. Activation of resident macrophages induces a state of inflammation at sites of infection. This diagram illustrates how resident macrophages, upon detecting infection, initiate an inflammatory response by secreting signaling molecules known as cytokines. At the top, a macrophage detects pathogens and responds by releasing several key cytokines. Interleukin-1 beta and tumor necrosis factor alpha increase the permeability of nearby blood vessels, which allows immune cells and helpful molecules in the bloodstream to enter the infected tissue. Interleukin-6 acts on fat and muscle cells, instructing them to increase metabolism, which generates heat and raises the temperature in the infected tissue, part of the fever response. Chemokine ligand A, another molecule secreted by macrophages, recruits neutrophils, important white blood cells, from the bloodstream and directs them to the infection site. Interleukin-12 recruits and activates natural killer cells, a specialized immune cell type, which then release additional cytokines that amplify the macrophage response. Altogether, these actions create a localized state of inflammation that is critical for containing and resolving the infection. NOD-like receptors recognize bacterial degradation products in the cytoplasm. This image illustrates how specific receptors inside immune cells, called nucleotide-binding oligomerization domain receptors, detect remnants of bacterial breakdown within the cell's interior. These bacterial fragments are released after the breakdown of pathogens inside a compartment known as the phagolysosome. Once inside the cytoplasm, these bacterial components bind to the intracellular receptors. Upon binding, the receptors interact with a signaling molecule called receptor interacting serine/threonine protein kinase 2. This interaction leads to a cascade of signaling events, including the activation of a kinase known as transforming growth factor beta activated kinase 1, which in turn activates another protein complex called the inhibitor of nuclear factor kappa B kinase. This complex then modifies a protein that normally holds back a powerful transcription factor, nuclear factor kappa B. Once freed, this transcription factor enters the cell's nucleus and turns on genes responsible for initiating inflammation. This process helps the immune system respond more effectively to invading bacteria. This image shows the early steps in how immune cells detect bacterial invasion inside the cell. After a bacterium is engulfed and broken down inside a compartment called a phagolysosome, small fragments of the bacterium escape into the cytoplasm. These bacterial fragments are recognized by special sensors inside the immune cell, known as nucleotide-binding oligomerization domain receptors. These sensors bind the bacterial fragments and then interact with a signaling protein called receptor interacting serine/threonine protein kinase 2. This interaction sets off a chain of signals that ultimately alert the immune cell to the presence of bacteria and begin the process of initiating an inflammatory response. This image continues the signaling cascade triggered by bacterial recognition inside an immune cell. After the bacterial degradation products activate the internal sensors, these sensors partner with signaling proteins. This interaction leads to the activation of a key enzyme that adds phosphate groups to other proteins in a chain reaction. One of these phosphorylated proteins targets an inhibitor that normally keeps the transcription factor inactive. Once the inhibitor is broken down, the transcription factor is released and moves into the nucleus. Inside the nucleus, it binds to DNA and turns on the genes needed to produce immune proteins that help fight infection. The inflammasomes amplify the innate immune response by increasing the production of IL-1 beta. This image shows how a structure called the inflammasome helps amplify the body's innate immune response. The process begins when an immune signal binds to a receptor on the cell surface. This triggers events inside the cell that lead to the production of an inactive form of an inflammatory molecule. Meanwhile, another pathway causes a drop in potassium levels inside the cell and activates proteins that come together to form the inflammasome. This structure then activates an enzyme that cuts the inactive inflammatory molecule into its active form. The active version is released from the cell and helps spread the immune signal, boosting inflammation where it's needed. Neutrophils are dedicated phagocytes, and the first effector cells recruited to sites of infection. Inflammatory cytokines recruit neutrophils from the blood to the infected tissue. This image illustrates how inflammatory signaling molecules help bring neutrophils, a type of white blood cell, from the bloodstream into infected tissues. To do this, the neutrophils must attach to and pass through the blood vessel wall. The image shows 4 key adhesion molecules involved in this process. First, vascular adhesins on the blood vessel wall help slow neutrophils down. Then selectins on the neutrophils help them roll along the vessel wall. Integrins on the neutrophil surface strengthen their grip by binding tightly to immunoglobulin-like molecules on the vessel wall. This secure attachment allows neutrophils to exit the blood vessel and enter the infected tissue to fight pathogens. This image shows the journey of neutrophils as they leave the bloodstream and move toward the site of infection, guided by chemical signals. First, neutrophils are loosely tethered to the inner lining of the blood vessels by selectins, which allows them to roll along the vessel wall. These weak interactions slow them down. Next, inflammatory molecules like chemokines cause the neutrophils to bind tightly to the vessel wall by strengthening the interaction between integrins on the neutrophils and immunoglobulin-like molecules on the endothelial cells. Once firmly attached, the neutrophils squeeze between the cells of the blood vessel wall in a process called diapetasis. After exiting the bloodstream, the neutrophils migrate through tissue toward the infection site, guided by chemokines like CXCL8. The inset on the top right shows a microscopic image of a neutrophil in the process of crossing the blood vessel wall. This entire process allows neutrophils to reach infected tissues quickly and launch an effective immune response. This illustration shows the beginning of neutrophil recruitment to sites of infection. Neutrophils travel through the bloodstream and must first slow down to exit the blood vessels. This slowing is achieved by a process called rolling adhesion. In the blood vessels, the inner lining of endothelial cells expresses selectin molecules on their surfaces. These selectins weakly bind to specific sugars on the neutrophil surface called sialyl Lewis X. Because the bonds are weak, neutrophils do not stick firmly, but instead roll along the vessel wall as they are pushed by the flow of blood. This rolling movement brings neutrophils close enough to receive further signals from the inflamed tissue, which will later lead to firm adhesion and migration out of the blood vessel toward the site of infection. This image continues the journey of a neutrophil as it leaves the bloodstream to reach an area of infection. After rolling along the endothelial wall, the neutrophil is exposed to signaling molecules known as chemokines, which bind to specific receptors on the neutrophil surface. These chemokines activate integrin proteins on the neutrophil, such as lymphocyte function-associated antigen 1, allowing it to bind tightly to adhesion molecules like intercellular adhesion molecule 1 on the endothelial cells. This firm attachment halts the neutrophil's rolling motion. Next, the neutrophil squeezes between the endothelial cells in a process called diapetasis. It moves through the basement membrane and into the infected tissue. Once inside, it follows a trail of chemokines deeper into the tissue, guided to the exact location of infection or injury. This step-by-step recruitment ensures neutrophils are delivered precisely where they are needed to fight pathogens. This image shows a high-resolution electron micrograph of a neutrophil captured as it moves through the endothelial barrier. The neutrophil's multilobe nucleus is clearly visible, surrounded by numerous granules that are packed with enzymes and antimicrobial substances. The large blue arrow points to the area where the neutrophil is actively pushing through the blood vessel wall, undergoing diapetasis. This process allows the neutrophil to exit the bloodstream and enter the tissue space, where it will follow chemical signals to the site of infection or injury. This real-life cellular snapshot emphasizes how the immune system precisely coordinates cellular movement, guiding neutrophils out of circulation and directly into areas where their pathogen-fighting abilities are urgently needed. This diagram illustrates how inflammatory cytokines help guide neutrophils to sites of infection by triggering a signal transduction cascade through G-protein-coupled receptors on the surface of the neutrophil. In the first panel, a chemokine binds to its corresponding receptor on the neutrophil membrane. This receptor is associated with a G-protein complex composed of 3 subunits. In its inactive state, the G-protein holds a molecule of guanosine diphosphate. In the second panel, upon chemokine binding, the receptor activates the G-protein by replacing guanosine diphosphate with guanosine triphosphate. This switch energizes the G-protein and primes it for action. In the third panel, the activated G-protein breaks apart into 2 functional pieces. These pieces travel through the cell's interior, setting off different signaling pathways. Ultimately, this cascade leads to altered gene expression, which equips the neutrophil to migrate efficiently, respond to chemotactic gradients, and perform its role in resolving infection. Neutrophils are potent killers of pathogens and are themselves programmed to die. This illustration shows the potent role that neutrophils play in the immune response to invading pathogens. Neutrophils are armed with a variety of receptors that allow them to detect and bind to many different bacterial and fungal molecules. These include receptors for lipopolysaccharide, sugars on the surface of pathogens, formylpeptides found in bacterial proteins, and molecules involved in complement activation. Once a neutrophil identifies a target, it engulfs the pathogen into a vesicle. Inside the neutrophil, this vesicle fuses with granules that contain toxic enzymes and antimicrobial substances. These substances rapidly degrade and destroy the ingested microbe, ensuring that the infection does not spread. After completing their task, neutrophils often undergo programmed cell death to limit tissue damage and signal for cleanup by other immune cells. This image illustrates the life cycle of a neutrophil as it attacks and eliminates a bacterial threat. The process begins when a neutrophil engulfs a bacterium, forming a membrane-bound compartment known as a phagosome. This phagosome then fuses with 2 types of granules inside the neutrophil: azurophilic granules, which contain antimicrobial substances, and specific granules, which contribute to the killing process. As the phagosome matures, its internal environment changes. The pH initially rises to activate certain antimicrobial enzymes and help kill the bacteria. Later, the pH decreases when the phagosome fuses with lysosomes, allowing acid hydrolases to break down the bacterium completely. After completing its mission, the neutrophil undergoes programmed cell death in a process known as apoptosis. Its remains are then safely cleared away by macrophages, which prevents tissue damage and helps maintain immune system balance. This diagram shows how neutrophils use reactive oxygen species to destroy pathogens. When a neutrophil encounters a threat, an enzyme called NADP oxidase converts oxygen into superoxide, a reactive molecule. This reaction is part of what's known as the respiratory burst. Next, the superoxide is transformed into hydrogen peroxide by another enzyme, superoxide dismutase. Hydrogen peroxide itself is harmful to microbes and helps kill them inside the phagosome. To protect surrounding tissues, neutrophils use the enzyme catalase to break down hydrogen peroxide into water and oxygen once its job is done. These tightly regulated chemical reactions are part of what makes neutrophils such effective killers, but they also contribute to tissue damage if left unchecked, which is why neutrophils are short-lived and programmed to die after their mission. This image highlights two key aspects of how neutrophils attack and eliminate pathogens. On the left side, a scanning electron micrograph captures the intricate web of neutrophil extracellular traps, often called nets. These are made of DNF fibers expelled by neutrophils as they die. The nets trap and immobilize bacteria, preventing their spread and making them easier to kill. On the right side, a fluorescence microscopy image reveals neutrophils in a tissue environment. The green and red staining likely marks specific immune components, possibly the DNA and granule contents released during net formation, while the black regions might be areas devoid of signal, such as vacuoles or degraded tissue. The vivid colors emphasize just how active and lethal neutrophils are in infected tissue. Together, these images demonstrate that even in death, neutrophils serve a purpose. They sacrifice themselves not just to engulf pathogens, but to create a hostile environment for microbes, buying time for other immune cells to arrive and finish the job. This chart lists the microorganisms that neutrophils are especially effective at eliminating. Among fungi, Aspergillus fumigatus is a key target. This is a mold that can cause serious lung infections, especially in immunocompromised individuals. The majority of the chart is dedicated to bacteria, showing the breadth of neutrophil defense. These include well-known pathogens like Staphylococcus aureus, which can cause skin infections and pneumonia, and Escherichia coli, often involved in gastrointestinal and urinary tract infections. Also listed are more opportunistic and resistant microbes such as Burkholderia cepacia, Mycobacterium fortuitum, and Clostridium difficile, the latter notorious for antibiotic-associated diarrhea. Altogether, this list highlights the critical role of neutrophils in combating a wide range of pathogens, from common culprits to those that challenge even modern antibiotics. When neutrophils function properly, they stand as a first line of defense, keeping many of these dangerous invaders in check. Inflammatory cytokines raise body temperature and activate the liver to make the acute phase response. This diagram illustrates the systemic effects of inflammatory cytokines, specifically interleukin-1, IL-1, interleukin-6, IL-6, and tumor necrosis factor alpha, TNF-alpha, as they coordinate the body's acute phase response to infection. These cytokines act on several organs and tissues to produce a comprehensive immune reaction. In the liver, IL-1, IL-6, and TNF-alpha stimulate the synthesis of acute phase proteins such as C-reactive protein and mannose-binding lectin. These proteins play a critical role in the immune response by activating the complement system and enhancing opsonization, a process that tags pathogens for destruction by immune cells. In the bone marrow endothelium, these cytokines promote the mobilization of neutrophils into the bloodstream. This mobilization supports phagocytosis, allowing neutrophils to reach infected tissues and engulf pathogens. The hypothalamus responds to the same cytokines by increasing the body's core temperature, resulting in fever. This elevated temperature helps to inhibit the replication of viruses and bacteria, creating an environment less favorable for pathogen survival. Meanwhile, in fat and muscle tissues, protein and energy stores are mobilized to generate the heat needed to maintain this increased body temperature. This metabolic shift not only supports fever, but also contributes to a broader antimicrobial environment by further reducing viral and bacterial replication. Together, these orchestrated responses highlight how cytokines mediate a systemic reaction to infection, aiming to contain and eliminate the invading pathogens efficiently. This figure provides an overview of acute phase proteins and their associated functions during the body's response to infection or injury. C-reactive protein, mannose-binding lectin, and lipopolysaccharide-binding protein are primarily involved in pathogen recognition. These proteins detect specific molecular patterns on microbial surfaces, facilitating the immune system's ability to identify and respond to invading organisms. Complement components such as C3, C4, C9, and factor B contribute to pathogen elimination. Once activated, the complement system enhances the ability of antibodies and phagocytic cells to clear microbes and damaged cells, promotes inflammation, and attacks the pathogen's plasma membrane. Granulocyte colony stimulating factor, serum amyloid A, and secreted phospholipase A2 are linked to the inflammatory response. These molecules help mediate inflammation by recruiting immune cells to sites of infection and modulating their activity. Lastly, fibrinogen, plasminogen, and tissue plasminogen factor are involved in coagulation. These proteins help form clots to contain infection and initiate wound healing, while also contributing to tissue repair and limiting pathogen spread. Together, these acute phase proteins form a coordinated defense system that detects, contains, and helps eliminate infections while promoting healing and homeostasis. This figure illustrates how the concentrations of various acute phase proteins in the plasma change over time following a moderate inflammatory stimulus. The graph reveals that proteins such as C-reactive protein and serum amyloid A respond rapidly and dramatically, with their plasma levels increasing by several 100% within the first few days. This swift elevation reflects their role in the early recognition and response to inflammation. Complement component C3 also rises early, but its increase is more modest and sustained over a longer period, indicating its ongoing role in pathogen elimination. Fibrinogen shows a slower but steady rise, peaking around 1 to 2 weeks post-stimulus, aligning with its function in coagulation and tissue repair during the later stages of inflammation. In contrast, albumin demonstrates a slight decrease in plasma concentration following the inflammatory stimulus. This drop reflects the liver's shift in protein production priorities during the acute phase response, favoring the synthesis of inflammatory proteins over housekeeping ones like albumin. Overall, the dynamic changes depicted in this figure illustrate the highly coordinated nature of the acute phase response, involving the rapid upregulation of key immune proteins and the temporary downregulation of others to restore homeostasis and fight infection. This image presents a structural depiction of a C-reactive protein pentamer, a key acute phase protein produced by the liver during the acute phase response. The colorful ribbon diagrams represent individual subunits of the pentameric complex, each forming part of a circular assembly. When inflammatory cytokines such as interleukin-1, interleukin-6, and tumor necrosis factor alpha signal the liver, hepatocytes respond by synthesizing this protein. C-reactive protein plays an essential role in pathogen recognition by binding to phosphocholine on the surface of dead or dying cells and certain bacteria, marking them for destruction by immune cells and activating the complement system. Its distinct pentameric shape is not only critical for its function, but also allows it to bind multiple ligands simultaneously, enhancing its efficiency during immune responses. The lectin pathway of complement activation is initiated by the mannose-binding lectin. This illustration depicts the mannose-binding lectin complex, which plays a central role in the lectin pathway of complement activation. The blue structures represent the mannose-binding lectin itself, arranged in a bouquet-like configuration that allows it to bind multiple carbohydrate surfaces of pathogens simultaneously. Attached to this lectin are the mannose-binding lectin-associated serine protease, MASP1 and MASP2, represented by the green circles. These enzymes remain inactive until the mannose-binding lectin binds to repetitive mannose residues on the surface of a pathogen, at which point MASP2 becomes enzymatically active. Once activated, MASP2 cleaves complement proteins C4 and C2, initiating a proteolytic cascade that results in the formation of the classical C3 convertase. This event marks the beginning of the complement cascade, which promotes opsonization, inflammation, and the eventual destruction of the pathogen. This figure shows how the mannose-binding lectin pathway is activated as part of the acute phase response during infection. When bacteria invade the body, they trigger macrophages to release interleukin-6, a cytokine that travels to the liver. In response, hepatocytes in the liver increase the production of several acute phase proteins, including mannose-binding lectin, C-reactive protein, and fibrinogen. Mannose-binding lectin targets carbohydrates commonly found on the surface of bacterial cells, but not on human cells. Once bound, it acts as both an opsonin, enhancing the uptake of pathogens by phagocytes, and a trigger for the complement system. Similarly, C-reactive protein binds to phosphocholine on microbial membranes, serving the same dual purpose. It marks pathogens for phagocytosis and activates the classical complement pathway. Together, these acute phase proteins amplify innate immune responses by promoting pathogen clearance and inflammation. This figure describes the step-by-step activation of the lectin pathway of the complement system, which is triggered when mannose-binding lectin binds to carbohydrates on the surface of a pathogen. The pathway begins when the enzyme MASP2, which is associated with mannose-binding lectin, becomes activated. MASP2 first cleaves complement protein C4 into two fragments, C4A, which is released, and C4b, which covalently attaches to the microbial surface. Then MASP2 also cleaves complement protein C2 into C2A and C2b. The C2A fragment binds to surface-bound C4b, forming a C4b2 complex. This complex acts as the classical C3 convertase. The C3 convertase, C4b2, then binds to complement protein C3 and cleaves it into C3A and C3b. C3b binds covalently to the microbial surface, effectively tagging it for destruction by phagocytic immune cells. This process enhances opsonization, inflammation, and ultimately the clearance of pathogens from the body. This figure illustrates the two main types of C3 convertase enzymes that are central to the complement activation pathways: the classical and the alternative. In the classical pathway, the C3 convertase is composed of two protein fragments, C4b and C2a, which combine on the surface of a pathogen to form the enzyme complex known as C4b2a. This enzyme is responsible for cleaving complement protein C3 into C3a and C3b, leading to further immune activation. In the alternative pathway, the C3 convertase is formed from the binding of Bb to C3b on the pathogen surface. This enzyme complex, called C3bBb, also cleaves C3 into C3a and C3b, amplifying the immune response. Both pathways ultimately lead to the opsonization of pathogens, inflammation, and recruitment of immune cells to the site of infection. C-reactive protein triggers the classical pathway of complement activation. This figure illustrates the initiation of the classical pathway of complement activation by the protein complex C1, which is composed of C1q, C1r, and C1s. On the left, the diagram shows the molecular structure. C1q acts as the recognition component with 6 arms, resembling a bouquet. Attached to it are two molecules, each of the protease C1r and C1s, which are responsible for enzymatic activity. On the right, an electron micrograph reveals the actual structure of C1q, visualized as flower-like shapes. When C-reactive protein binds to phosphocholine on a pathogen surface, it provides a docking site for C1q. This binding activates C1r, which then activates C1s, beginning a proteolytic cascade that amplifies the immune response through opsonization, inflammation, and cell lysis. This figure illustrates the initiation of the classical pathway of complement activation by the protein complex C1, which is composed of C1q, C1r, and C1s. On the left, the diagram shows the molecular structure. C1q acts as the recognition component with 6 arms, resembling a bouquet. Attached to it are two molecules, each of the protease C1r and C1s, which are responsible for enzymatic activity. On the right, an electron micrograph reveals the actual structure of C1q, visualized as flower-like shapes. When C-reactive protein binds to phosphocholine on a pathogen surface, it provides a docking site for C1q. This binding activates C1r, which then activates C1s, beginning a proteolytic cascade that amplifies the immune response through opsonization, inflammation, and cell lysis. Toll-like receptors sense the presence of the 4 main groups of pathogenic microorganisms. This chart outlines how various toll-like receptors, or TLRs, recognize specific microbial products across the 4 main groups of pathogens: bacteria, viruses, fungi, and parasites. TLR1 and TLR2 form a heterodimer located on chromosome 4 and detect bacterial lipoproteins and parasitic components such as glycosylphosphatidylinositol, expressed on the plasma membranes of monocytes, dendritic cells, eosinophils, basophils, and mast cells. Similarly, TLR2 paired with TLR6 recognizes lipotecoic acid from gram-positive bacteria and zymosin from fungi, also at the plasma membrane. The exact ligands for TLR10 are still unknown, though it's present in a variety of immune cells including plasmacytoid dendritic cells and B cells. TLR4, encoded on chromosome 9, responds to lipopolysaccharide, or LPS, from gram-negative bacteria and is found on the plasma membrane of macrophages, dendritic cells, mast cells, and eosinophils. Within endosomes, TLR7 and TLR8, both responsive to single-stranded viral RNA and TLR9, which detects unmethylated CpG DNA from bacterial and DNA viruses, play key roles in antiviral defense, particularly in plasmacytoid dendritic cells, NK cells, and B cells. TLR3, also located in endosomes, recognizes double-stranded viral RNA and is especially active in NK cells. Lastly, TLR5, located at the plasma membrane of intestinal epithelial cells, detects bacterial flagellin, underscoring its role in maintaining gut immunity. This illustration demonstrates how different toll-like receptors are specialized to detect microbial products either outside the cell at the plasma membrane or inside the cell within endosomes. The toll-like receptor 1 and toll-like receptor 2 form a heterodimer and are located on the plasma membrane, where they recognize bacterial components such as lipoproteins. Similarly, toll-like receptor 4, which also resides on the plasma membrane, detects lipopolysaccharides found on the surface of gram-negative bacteria. Inside the cell, toll-like receptor 3 is found within endosomes. It is responsible for detecting double-stranded viral RNA, a hallmark of viral infections. This strategic localization enables the immune system to sense both extracellular and intracellular threats, initiating appropriate defense responses depending on where the microbial signature is detected. Genetic variation in toll-like receptors is associated with resistance and susceptibility to disease. This figure explains how genetic variation in toll-like receptors is associated with either resistance or susceptibility to disease, depending on their type and cellular location. On the left, a phylogenetic tree groups toll-like receptors based on the types of microbial molecules they recognize. Those that detect lipids, such as toll-like receptors 1, 2, 4, 6, and 10, are clustered together in a blue-shaded area. Receptors that detect nucleic acids, including toll-like receptors 3, 7, 8, and 9, form a separate group highlighted in pink. Toll-like receptor 5, which recognizes proteins such as flagellin, stands alone in the yellow-shaded section. On the right side, a bar graph shows how much genetic variation, represented by the symbol omega, each receptor exhibits. Toll-like receptors that are found in endosomes and sense nucleic acids, such as toll-like receptors 3, 7, 8, and 9, show less genetic variation, possibly due to evolutionary pressure to conserve their function and avoid autoimmunity. In contrast, cell surface receptors like toll-like receptors 1, 2, 4, 5, 6, and 10 exhibit more variation, possibly because they are under constant pressure from rapidly evolving extracellular pathogens. Together, these insights reveal how the location and microbial target of a toll-like receptor may shape its genetic diversity, influencing how individuals respond to infections. Internal detection of viral infection induces cells to make an interferon response. This diagram outlines how cells detect internal viral infections and initiate an interferon response to defend against them. The process begins when a virus replicates within the cytoplasm of a host cell, producing RNA molecules that are not capped and contain a distinctive 5-triphosphate group. These uncapped RNA molecules are recognized by receptors in the cytoplasm called Reg-like receptors. When these receptors bind to the viral RNA, they associate with an adapter protein called MOVs, which is anchored on the outer membrane of mitochondria. The interaction between the receptor and MOVs causes the receptor to dimerize or pair up, which triggers downstream signaling events. This leads to the recruitment of several other signaling proteins such as TRAF6, FADD, and TRAD, which in turn activate two key transcription factors, interferon regulatory factor 3, and nuclear factor kappa light chain enhancer of activated B cells. Interferon regulatory factor 3 stimulates the production and release of type I interferons, while nuclear factor kappa light chain enhancer of activated B cells promotes the secretion of inflammatory cytokines. Together, these molecules coordinate a powerful antiviral response by alerting neighboring cells and activating immune defenses to limit the spread of the virus. This image illustrates how a virus-infected cell initiates and amplifies an interferon response to alert neighboring cells and enhance antiviral defenses. Once a virus enters and replicates within a host cell, viral nucleic acids activate transcription factors, such as nuclear factor kappa light chain enhancer of activated B cells, activator protein 1, and interferon regulatory factor 3. Together, these factors induce the transcription and production of interferon beta. The interferon beta that is secreted from the infected cell binds to type I interferon receptors on the same cell in an autocrine manner, and on nearby uninfected cells in a paracrine manner. When interferon beta binds its receptor, it activates signaling pathways that lead to the expression of additional interferon-stimulated genes, including interferon regulatory factor 7. This transcription factor is crucial for the production of interferon alpha, which further enhances the antiviral state within the infected cell and in surrounding cells. The result is a robust interferon-mediated immune response that limits viral spread and primes uninfected cells for defense. This image summarizes the outcomes of the interferon response initiated by virus-infected cells. When a cell becomes infected by a virus, it detects the viral presence and begins producing interferon alpha and interferon beta, which are type I interferons. These molecules are secreted and bind to interferon receptors on the same cell and neighboring cells, triggering a coordinated antiviral defense. This interferon response leads to three major effects. First, it induces a state of resistance to viral replication in all nearby cells, thereby containing the spread of the infection. Second, it increases the expression of ligands on infected and surrounding cells that are recognized by receptors on natural killer cells. Lastly, it activates natural killer cells, empowering them to identify and destroy virus-infected cells more efficiently. This cascade of signaling events ensures a rapid and localized innate immune response to viral threats. Plasmacytoid dendritic cells are factories for making large quantities of type I interferons. This image shows a highly magnified view of a plasmacytoid dendritic cell, a specialized immune cell known for its exceptional ability to produce large quantities of type I interferons during viral infections. These cells are equipped with extensive internal membrane structures and a dense cytoplasm, reflecting their robust capacity for protein synthesis and secretion. Upon detecting viral nucleic acids, often through toll-like receptors located in endosomal compartments, plasmacytoid dendritic cells rapidly initiate transcription and translation of interferon alpha and interferon beta. These interferons are then secreted to alert neighboring cells and orchestrate a systemic antiviral response, making plasmacytoid dendritic cells central players in early innate immunity. Natural killer cells are the main circulating lymphocytes that contribute to the innate immune response. This figure illustrates the timeline of immune responses following a viral infection, highlighting the central role of natural killer cells as early responders. Shortly after infection begins, innate immune cells, such as dendritic cells and macrophages, produce key cytokines, including interferon alpha, interferon beta, tumor necrosis factor alpha, and interleukin 12. These cytokines activate natural killer cells, which peak in activity around days 2 to 4, where they play a critical role in killing virus-infected cells and helping to limit viral replication. As the innate response starts to wane, adaptive immunity takes over. Cytotoxic T cells become the dominant cell type responsible for killing infected cells, peaking after day 5. Meanwhile, the viral titer rises initially but begins to decline as natural killer cell and T cell responses become more effective. This chart underscores how natural killer cells form a vital bridge between the innate and adaptive immune responses, acting as swiftly before T cells are fully activated. Two subpopulations of NK cells are differentially distributed in blood and tissues. This figure demonstrates the circulation and tissue distribution of two distinct subpopulations of natural killer cells, which are critical players in the innate immune system. These subtypes are differentially localized in blood and tissues. Natural killer cells are produced in the bone marrow and enter the bloodstream, circulating through the body. Some migrate into tissues like the skin, while others enter lymph nodes through specialized blood vessels called high endothelial venules. Within lymph nodes, they may encounter signals that influence their function and fate. These cells can exit the lymph node via the efferent lymphatic vessels and re-enter circulation. This movement ensures that natural killer cells are strategically positioned both in peripheral tissues, where infections may initiate, and within secondary lymphoid organs, where they interact with other immune cells. The figure highlights how the anatomical distribution of natural killer cell subpopulations supports both surveillance and rapid response to infection. NK cell cytotoxicity is activated at sites of virus infection. This figure illustrates the sequential process by which natural killer cells are activated to eliminate virus-infected cells at the site of infection. Initially, viral infection of tissue cells triggers the production of type I interferons, a family of signaling proteins that serve as early warning molecules. These interferons act on nearby natural killer cells, promoting their rapid proliferation. As the number of natural killer cells increases, type I interferons also drive their differentiation into cytotoxic effector cells, specialized in killing. Once fully activated, these effector natural killer cells recognize and induce apoptosis, a programmed form of cell death in the infected cells. This response limits viral replication and prevents further spread of the infection, representing a key early defense mechanism of the innate immune system. This figure compares the signaling pathways triggered by two different toll-like receptors, or TLRs, inside endosomes that detect viral RNA and lead to the production of type I interferons. On the left, toll-like receptor 7 recognizes single-stranded RNA within endosomes. Upon detection, it signals through the adapter protein myeloid differentiation primary response 88 or mid-88, which activates interferon regulatory factor 7 or IRF7 via the kinase TBK1. This signaling cascade promotes transcription of both interferon alpha and interferon beta genes, resulting in the production and secretion of these antiviral proteins. On the right, toll-like receptor 3 detects double-stranded RNA, also a viral signature, and instead signals through the adapter protein TRIF, or TR domain containing adapter-inducing interferon beta. This activates IRF3 through kinases TBK1 and IKK epsilon, which leads to the transcription of only the interferon beta gene. In both cases, the interferons produced act to initiate antiviral responses in neighboring cells and amplify the immune defense. NK cells and macrophages activate each other at sites of infection, This figure shows how natural killer cells and macrophages activate each other at the site of a viral infection. When a macrophage becomes infected by a virus, it begins to secrete signaling molecules such as the chemokine CXC motif ligand 8 and the cytokine interleukin 12. These molecules attract and stimulate natural killer cells. When a natural killer cell comes into contact with the infected macrophage, the two cells form a close connection called a synapse. Through this synapse, the macrophage delivers more activating signals using interleukin 12 and another cytokine called interleukin 15. These signals cause the natural killer cell to multiply and develop into an effector natural killer cell. The activated natural killer cells begin to produce and release a signaling molecule called interferon gamma. Interferon gamma binds to receptors on the macrophage, further enhancing its activity. This causes the macrophage to increase its ability to engulf and destroy pathogens and to secrete more inflammatory signaling molecules. This back and forth communication helps coordinate a strong early immune response to viral infection. Interactions between dendritic cells and NK cells influence the immune response. This image illustrates the dynamic interplay between dendritic cells and natural killer cells, which determines the direction of the immune response. In the left panel, immature dendritic cells activate natural killer cells by producing interleukin 15, promoting their proliferation and differentiation into effector cells capable of killing virus-infected cells and secreting cytokines. In the middle panel, when natural killer cells are abundant and outnumber dendritic cells, they exert regulatory control by killing the dendritic cells, thereby suppressing further dendritic cell function and limiting immune escalation. In the right panel, when natural killer cells are scarce and dendritic cells are more numerous, the dendritic cells are not killed but instead are guided by the natural killer cells to mature. These mature dendritic cells then migrate to lymph nodes and initiate the adaptive immune response by activating T cells. Thus, the relative numbers and functional states of these cell types determine whether the immune system continues to rely on innate responses or shifts toward activating adaptive immunity.